Alright everyone, it's time to talk about one of the most important topic of the recombinant DNA technology, that is the difference between two important molecular vector, that is bacterial artificial chromosome and yeast artificial chromosome. People generally think like it's artificial chromosome, so let's compare BAC versus YAC, bacterial artificial chromosome versus yeast artificial chromosome. But actually there's a lot of difference between BAC and YAC. YAC is actually used Yeast artificial chromosome actually used to clone larger or lengthier fragment of the DNA in eukaryotic host, where the BAC or bacterial artificial chromosome can have a higher or larger insert size, but the cloning will be done inside the bacterial system or prokaryotic system only. That's the major difference. So let's look at the difference. In the left hand side we have yeast artificial chromosome, right hand side bacterial artificial chromosome. So the very first thing is that yeast artificial chromosome are genetically engineered chromosomes derived from the DNA of yeast Saccharomyces cerevisiae, where bacterial artificial chromosome, an engineered DNA molecule used to clone DNA fragment in the bacterial cell. This is what we just said. This is what I just said in the last slide. YACs are used to clone very large DNA fragments, 1000 to 2000 kilobases long DNA fragment, while bacterial artificial chromosome can hold up to 300 kilobases of DNA fragment. The yeast artificial chromosome are inefficient in many ways because of the larger length of the DNA that it carries. The process of transfer is inefficient. Both of them are not that much efficient like plasmid, like bacteriophage, like cosmid. No, unlike bacterial artificial chromosome, it is not so hard to construct yeast artificial chromosome library, but bacterial artificial chromosome library construction is very difficult. So if our aim is to produce a genomic DNA library or cDNA library, we generally take help from the bacteriophage as a vector, or phagmid or cosmid as a vector, rather than bacterial artificial chromosome. Although bacterial artificial chromosome can carry lengthier fragment of the DNA, still it is not used because it's very difficult to construct a library with bacterial artificial chromosome. But yet yeast artificial chromosome has less difficulty in constructing a genomic DNA library. The yeast artificial chromosome are relatively less stable than the bacterial artificial chromosome. Yeast artificial chromosome are unstable, obviously, because it's a artificially engineered chromosome. It has less stability. And another thing is that these artificial chromosomes tend to contain scrambled inserts, that is composites of DNA fragments from more than one site, while the bacterial artificial chromosome contains a pure insert that is taken from a single stretch of the bacterial genome. The linear yeast artificial chromosomes, which tend to break under shearing forces, while the bacterial artificial chromosome are circular, and circular or coiled bacterial artificial chromosome can resist the breakage. That is another difference between the two. Yeast artificial chromosome are hard to isolate from yeast cell, while bacterial artificial chromosome are easy to isolate. And one more thing is that the fundamental difference is that in yeast artificial chromosome, as I said, it's a linear one. It has a centromere and two telomeres in the surrounding place, while bacterial artificial chromosome, circular in nature, sometimes also coiled in some way, that can resist the breakage and can also be easy to isolate. But in case of yeast artificial chromosome, they are difficult to isolate. Okay, so yeast artificial chromosome is only used in situations where we need to check the expressivity of a particular gene in an eukaryotic system. In that case, we first cut the gene of interest, clone it and insert it inside the yeast artificial chromosome, insert it inside the target eukaryotic organism, that is yeast in this case, and then we'll check the expressivity. Then we'll check the behavior of the particular gene inserted. That's what the yeast artificial chromosome is used for. But bacterial artificial chromosome is used again to hold a huge length of the DNA. But again, practically speaking, bacterial artificial chromosome is less used, although yeast artificial chromosome is used in some case, but bacterial artificial chromosome is generally not used. We use bacteriophage lambda as a vector, we use phagmid as a vector, we use the plasmid widely as a vector. So that is all about the difference between the yeast artificial chromosome and the bacterial artificial chromosome. If you like this video, please hit the like button, share this video with your friends and colleagues, and subscribe to this channel to get more videos like that in future. Thank you. Bye. Okay everyone, it's time to talk about another very interesting concept of recombinant DNA technology chapter, and that is Genomic DNA Library. Now I want to keep this lecture simple and straightforward. What is Genomic DNA Library? DNA library. What is this term? DNA library. Now, what library is? Library is a repository, right? It's a composite of materials. So let's say library of books. It's a composite of books. Books from different genre, books from different subjects. They are kept in different rows and columns of different arrangement in the library. So basically, library is a construct made from every information that are available regarding, let's say in case of book library, regarding a book. But when we say genomic DNA library, it should carry all the information regarding every single gene that are available inside a genome of a whole organism. That is known as a genomic DNA library. So when you construct a library of genome, so genome means any particular gene under any particular gene located in any particular chromosome of that organism, or any particular portion of the genomic DNA of that organism if it's prokaryotes. That is known as a every single gene component is under the term genome. So all those genes are extracted, cut out from the genome, and they are cloned and they are stored in a genomic DNA library, so that in future times, whenever we want to work with any of that, any one of those, if you want to work with it, then we can easily isolate that. We can easily pick that particular clone with our target DNA. We can pick that clone with our target DNA. We can pick that clone with the help of our target. There are multiple systems to screen the colony of bacteria that carries the gene of our interest. We can find it out. Okay, we do that. That is genomic DNA library. So let's say this is so there are these all plates. Okay, so all plates, petri plates, where we have different bacterial colonies. We know that there is a marking that if this is a whole genome, that this segment of the genome is stored here, this segment of the genome is stored here, this segment stored here, let's say this segment stored here. Okay, and the colony that formed also, they have different color, and based on the color we can select whether our interest, whether the colony, the bacterial cell there carries the gene of our interest or not. We can also find it out. Okay, so genomic DNA library, in simple word, contains DNA fragments representing the entire genome of an organism. Let's say we are making a genomic DNA library of a mouse. So we will go for every single gene that is present in the mouse chromosome, all the locations of the mouse chromosome, and every little gene, every single gene, will select that, will cut it out from that mouse chromosome, and individually those segments of the genes are cloned and they are stored as this colony of bacterial culture in petri plates. Basically, because in this case, creating a library and storing means what? It's a live culture. The bacterial cells are alive, so we need to continue the culturing process to keep it alive. Okay, so imagine in this case of construction of a genomic DNA library, there are few things. First of all, the whole genome need to be identified, all the genes need to be identified, there which genes will clone, then those those genes will become our target DNA of interest, right? Target DNA or gene of our interest. So we'll cut them, then we'll do the cloning of individual section of the individual fragment of the DNA that we cut. So it may carry one gene, more than one gene, obviously more than one genes, more than two genes it may carry, but we fragmentize that. So we do the fragmentation randomly, or sometimes we do the restriction endonuclease. So after the fragmentation is done, then the fragmented DNA will be tagged, recombinant DNA will be formed. What we will use here is vector, molecular cloning vector. The molecular cloning vector that we use will now carry the fragmented target DNA, which will be cloned there. Alright, so after the cloning is done, then the recombinant plasmid or the recombinant vector, not only plasmid, basically in case of genomic DNA library construction we don't use plasmid, we use the vector which has larger insert size, that is generally bacteriophage lambda or phagmid or cosmid, we can use any of that. So once that vector carries the target DNA, so we call it recombinant plasmid or recombinant vector. So that recombinant vector is then allowed to transfer the target DNA along with the rest of the vector material inside the host cell. So there the host is bacteria. Some cases the host can be eukaryote, but in this case the host is bacterial cell, let's say E. coli. So what is the process of gene or genomic DNA library construct? See, this is the human DNA, approximately 3 into 10 to the power 9 bases long. Okay, and what we do, a partial digestion with a restriction endonuclease Sau 3A into 20 kb fragments. Also, it's a huge fragment. You can see that 3 into 10 to the power 9 base pair long fragment, but we do random fragmentation with a treatment with SAU 3A or any particular restriction endonuclease here. We, for example, is SAU 3A. So that generates approximately 20 kb length fragments with sticky ends. Sticky end means basically, let's say this is what sticky end means. So this is how the sticky end looks like. One of the strand has overhang structure like this. This is known as sticky end. This is where they are attached with the help of hydrogen bonds, the two strands of the DNA, and both the terminal site single strand of the DNA sticking out. This is known as sticking end. So sticky end is generated with 20 kb fragment, and you know the phage vector can easily carry 20 kb length of the DNA. So this is the bacteriophage lambda, okay, and this is a replaceable region. We know that the lambda phage have two different type, insertion vector and replacement vector. Let's say this is a replacement vector. We will replace the replaceable region with the help of 20 kb fragment with sticky end. We attach 20 kb fragment with sticky end there. So once that is done, the ligation will be done, okay, with the help of the sticky end, because the moment we treat the bacteriophage with a restriction endonuclease, that is BAM-H1, that will remove the replacement region or discard the any replaceable region, and it also develops a sticky end in the lambda phage DNA. So lambda phage DNA or vector DNA has a sticky end. A 20 kb fragment of our human genome or human DNA is also having a sticky end. Now this sticky end will stick together. That means basically sticking means your hydrogen bonding between the single strands of the DNA. So this bonding is done and the bonding is made. So now here we can see yellow and green. Green means human DNA, yellow is the bacteriophage DNA, the vector DNA. So this is a hybrid DNA, or we can call as a recombinant DNA, recombinant lambda DNA. Okay, and this recombinant lambda DNA is ready. The recombinant lambda DNA will be packaged inside the lambda phage head by the phage itself. Now this phage is ready to infect a bacteria, which is the phage's host. So this phage will now infect a bacteria, transfer this particular recombinant DNA inside the bacteria, and then the bacteria will be allowed to grow in a culture media. And among the bacteria, there are different processes. The phage can either infect the bacteria and deliver the target recombinant DNA there, or there is the possibility that the phage without the recombinant DNA can infect. So in that case, the recombinant DNA cannot enter the bacteria. So there are these options. Either the host cell, that is bacteria, can receive the recombinant DNA where our DNA, target DNA, that is the human genome DNA, is inserted, or a situation where there is no target DNA inserted. Both are equally possible. So there are two types of host cell. One type of host cell received the target DNA, that is the human DNA, and another type they don't receive any target DNA. So this is a very important step of Genomic DNA Library, that is known as Genomic DNA Library screening. Screening means filtration or filtering. Filtering for what? Or screening for what? Screening for distinguishing the host cell with or without our DNA of interest, that is the human DNA fragment. So if our host cell, that is E. coli, lacks the human DNA fragment, then we don't take those colonies, we don't select those colonies, because we don't need them. We only need those colony of bacteria who successfully received the target DNA, that is 20 kb fragment of human DNA in it. Then only we will select those colonies only. And not only we select those colonies, we allow those colonies to grow and divide and make more and more cells. So we'll culture those colonies, subculture those colonies, those plates multiple times, and we'll maintain that as a genomic DNA library. That is the idea of maintenance of genomic DNA library. So first of all, obviously, there's a construction of genomic DNA library where we need the target DNA, where we need the molecular cloning marker, and where we need the host cell. And then the second step is screening of the host cell, whether that host cell carries the gene of our interest or not. And the third thing is maintenance of the genomic DNA library. Construction, screening, maintenance. These are the three important steps of a genomic DNA library. So here is another example. Let's say genomic DNA library of human cells. So isolated DNA is already isolated. These are plasmid vector, for example. Now obviously the whole genome is huge. Eukaryotic genome, let's say human DNA, human genome is huge. So we need to cut it down. We can cut it randomly, or what we can say, we can cut it in fragments utilizing the restriction endonuclease. we can use restriction endonuclease to cut it. Random digestion, or what you can say as a partial digestion, not foolproof digestion, partial digestion can be made. So the digestion is done with restriction enzymes. Restriction enzymes can be used. In this case, the restriction enzyme SAU3A was used earlier. So use that restriction enzyme, but basically we need to use a similar restriction enzyme, or restriction enzyme, either same restriction enzyme or restriction enzyme that can generate similar kinds of ends. So in this case, what we are saying is that the restriction endonuclease that we need to use, the restriction endonuclease can be either same restriction endonuclease used for both, for cleaving the target DNA as well as the vector, or we need to use a similar restriction endonuclease that will generate similar cut ends. So for example, in this case you can see that we generated 20 kb fragment with sticky end. If we cut the human DNA with SAU3A, if we cut the bacteriophage DNA with BAMH1, it will also generate similar overhang with complementary sequence, so that our target DNA with sticky end can match with the sticky end of the lambda-phage DNA, and the recombinant plasmid recombinant vector can be created. The similar thing happens here. So we will cleave this target DNA of ours, partially digest that, and also we'll cleave the plasmid vector, and then we use the plasmid vector as well as we use the cleaved DNA to be recombined with the plasmid vector. Once the recombination is done, then the recombinant plasmid will be transferred or inserted inside the bacterial cell, and once it's inserted inside the bacterial cell, then the bacterial cells will be allowed to grow. Okay, and then the amplification, this is known as amplification of the gene library. So basically this is the transfer of the recombinant DNA. So when we say recombinant DNA technology, it includes all the techniques used in the formation of recombinant DNA, conversion of recombinant DNA, transfer of recombinant DNA inside the host, and maintenance of the host cell. So the recombinant DNA is transferred into the host cell, which is E. coli here, and we allow the E. coli to grow and divide. We allow its amplification. Okay, so genomic DNA library amplification is done, and once that is done, then obviously we can isolate those bacteria and we can easily separate any particular DNA. If you want to work with any particular fragment of DNA of our target, then what we can do is directly refer to a particular colony in a plate, and we know we can mark the plates that this plate contains a gene number this to this, X to Y, then another one is a Y to Z, so like that. So we can directly go there, we can isolate that colony, the bacteria from the colony, and we can easily work with the target DNA of our choice. So that's why the genomic DNA library is created, because it is basically a picture of every single gene that is available, that is present in a particular organism. Okay, that's why the genomic DNA library is so important. Now there are multiple types of vectors used in genomic DNA library or construction of a genomic DNA library. We know that the vectors that we use to construct genomic DNA library must have a larger or lengthier insert size. A large insert size is really important, because the DNA fragments, although we can fragmentize into 20 kb, 30 kb, 35 kb, but still we need to keep it short till 30-35 kb, because we want to clone the fragment of target DNA, whether it's eukaryotes or prokaryotes, doesn't matter. The fragment of target DNA, we need to clone it using either the phage, bacteriophage as a vector, or phagemid as a vector, or cosmid as a vector. So in all these vectors, they have a maximum capability of insert size of 35 kilobases. So we need to reduce, we need to keep the fragment of our gene of interest or target under the 35 kb length, that we can achieve easily with the treatment of restriction endonuclease to the target genome. So what are the vectors that we use here? See, the vector plasmid is used, the host is E. coli, and the insert size of plasmid can carry 5 to 25 kilobases. If we use lambda-phage, again the host is E. coli, the insert size 35 to 45 kilobases. P1 phage is used, host is E. coli, the insert size is 70 to 100 kilobases. Then we have PACs, host is E. coli, size is 100 to 300 kilobases. We have bacterial artificial chromosome, again host is E. coli, insert size is maximum 300 kilobases and less than 300 kilobases. Yeast artificial chromosome, the host is eukaryotic cell, that is Saccharomyces cerevisiae or yeast, the insert size is 200 to 2000 kilobases. So these are all different. And another is the human artificial chromosome, or HACS, HAC. The host is human cultured cell, which is difficult to maintain. The whole genome library construct with the human artificial chromosome is more difficult, more laborsome. But it can carry huge length of the DNA, that is more than 2000 kilobases of the DNA can be cloned using human artificial chromosome as a vector, but not used. The generally used vector for this process of construction of genomic DNA library is plasmid and lambda phage. These two are widely used one, and we also use yeast artificial chromosome for having a moderate to lengthy sequence to be cloned. So what are the problems of construction of genomic library and what are the solution towards it? So we'll talk about problems and solutions. The problem number one is the gene may be cut internally with the restriction enzymes used. Therefore, a single required sequence is not obtained. Now that's a possibility because the genome is of huge content, right? And we use restriction endonuclease to cleave it into smaller fragment. We don't know where exactly the restriction endonuclease is going to cleave it, but yeah, we can run bioinformatics tool to study, like we put the restriction endonuclease name and we also put the sequence. So from that we can get a data about approximate number of fragments that we get, the approximate number, the average size of the fragment that we get, the lowest size of the fragment that we can get, the highest size of the fragment that we can get. We can get about all this information utilizing bioinformatics tool these days. But still, sometimes the theoretical knowledge and practical process are a little different. While doing practically, the restriction endonuclease may cleave any particular gene of interest from middle. It shouldn't supposed to cleave there, but maybe due to some reason it cleaves, as a result of which the fragment that we wanted to generate failed to be generated. That's one problem. Second problem is that the gene of interest obtained by the restriction enzyme may be larger than the vector. Yeah, that is one really practical problem because the vector size is not big. See, the vector size is limited and the insert size that the vector can carry is also limited. But after the cleavage with restriction endonuclease, it can generate fragments which is more than the capability of the vector. Let's say we want to clone the whole genome and the fragment portion of the genome of human being utilizing bacteriophage lambda as a vector. Bacteriophage lambda has a capability of 35 to 45 kilobases long DNA fragment as a vector. So insert size as a vector. So now we cleave it with a particular restriction endonuclease and the fragment size, average fragment size is generated is, let's say, 60-70 kilobases. So 60-70 kilobases size of a fragment cannot be cloned using the lambda as a vector. So in that case, it's of no use. So in that case, we need to use or treat it with some restriction endonuclease that generates a manageable size of the insert. Okay, that's really important. Solution is that random cloning of DNA fragments of large size greater than 20 kb is needed. So basically we don't care about any particular gene. Earlier people thought when we are making the genomic library, and you can also think of the same way. Let's say this is the whole gene, whole DNA, the genome for example. This is a fragment, this is another section, another. So we can fragmentize that to 1, 2, 3, 4, 5 different fragments. And people thought that, yeah, we'll first cut this fragment 1, we'll clone it, fragment 2, we'll clone it, fragment 3, we'll clone it. We'll do that for all the other fragments like that, particularly individual fragment cloning. But now we know that this is not going to happen because there are some problems associated with it. So to get a solution is that any fragment which is more than 20 kb in length, 20 kb, more than 20 kb in size, which is considered a standard size for the genomic DNA library construction. In that case, instead of thinking of exactly borderline fragments to be cloned, we clone it randomly. So for example, instead of that, what is the approach that we took is that, let's say in the first fragment, random fragment, this is the fragment 1, second fragment was something like this, third fragment was something like this, fourth was something like this, fifth something like this, sixth something like this. So there are overlapping regions because it's a random fragmentation. There are overlapping regions that is created. Okay, so this is the approach that we take, and after we get multiple fragments with the overlapping sequences, it's fine, we'll clone all of them. After the cloning is done, when we need to work with them, we'll extract the DNA, isolate the DNA, we'll run a sequencing, and after running a sequencing, what we will do is simply we will align the sequence and we'll align it based on the overlapping region, and based on the overlapping region we can reconstruct the whole genome from that. That is really, really important. Reconstruction of genomic library from the data that we obtain after DNA sequencing, that changes the whole equation of genomic DNA library construction, because we first thought that we can construct it with targeted fragment size, which is not possible because of the restriction enzyme treatment here. It's not possible, it lives anywhere, it's not possible. So what we do here, what we did, instead of doing that, we do random fragmentation greater than 20 kb of fragment. And when we do random fragmentation, then we isolate the DNA, we sequence that, we try to find out and match the overlapping region. Once we find the overlapping region, the whole sequence can be reconstructed with the help of softwares and algorithms. That is what we do with genomic DNA library construction, as well as that's what we do in case of cDNA library construction. Now remember, I told you in genomic DNA library construction there are multiple stages. The first stage is construction, second one is screening, third one is maintenance. So this is the second step, that is screening. For the process of screening of genomic DNA library, there are multiple approaches. So basically screening means we are trying to find out the host cells, that is bacteria, with the target DNA of our choice. Okay, we need to find it out. So any of the bacteria that carries the target DNA of our choice, we are going to find it out. How do you do that? With different sorts of hybridization, like colony hybridization technique. We can use colony hybridization. We can use screening by DNA hybridization technique. We can use PCR to find out the target DNA inserted or not. We can use immunological assays, and we can also use protein function test for analyzing the target DNA's presence in the host cell. One such example is this hybridization assay. What we do is that cloning of the plate. So let's say this is the plate, the agar plate, and in this plate what we see the bacterial colony. Bacterial colonies that carry recombinant plasmids are present. What we will do here, we'll replicate this plate. How? We'll create a filter just in shape of the plate's diameter. We place it on top of the bacterial colony so that the bacterial colony get in touch with the filter. Okay, and we take a sample of the cells of the colony as it is printed into the filter, right? Just the filter is in touch with the colony, so anything in the colony that will be in touch, the bacteria in the colony will be in touch in the with the filter. Once that is done, then what we do, heat the filter paper to denature the DNA. We heat the filter paper to denature the DNA and wash the filter paper with radioactive probe, which will only attach to gene of interest. So basically what we did is that, let's say this is the paper and let's say this is one place. So when we heat the DNA, denatured, so only single stranded DNA is now present, and now we know that this is a target. Let's say this is the target DNA. Target DNA carries A, A, C, G, T. So the probe that we design would carry T, T, C, T, T, G, C, A. So this should be our probe, and the probe is added to radioactive dye or radioactive tag. So what we do is that we add the probe. Let me change the color for the probe. We add the probe which has complementary nature to that of our target DNA only, and with the radioactive tag. Now in this case the probe is not binding, probe bound here, but probe did not bind here. So the colony that is present here did not carry the target DNA, but here the colony is present. So our probe, which is complementary towards the target DNA region, bind properly, so radioactive signal will come from that point. So ultimately if we look at this plate, we'll see radioactivity coming from this side, but no radioactivity from this point, although there was a colony here. So the colony that was present there initially in the plate, if we now go back to the plate, because we did a replica plating, it's known as a replica plating, utilizing the paper. This was paper, this is the plate. So now if we go back to the plate, there is a colony here, there is a colony here. So the colony that was present here does not carry the gene of our interest. The colony here carries gene of our interest. This is a way to screen. So from this colony, from the plate, we found out which bacterial colony carries the DNA of our interest, the DNA of our choice. So now once we know that, we can directly go into the colony and you can easily pick cells from the colony. We can culture that, okay, liquid culture, and then we can extract the DNA and we can work with the DNA, we can sequence the DNA, and we can do other things with the help of this particular DNA. So this is the screening approach. Screening means basically finding out, fishing out the target DNA of interest from the rest of the colony. So this is the replica plating process. Okay, the replica plating process helps us to get to know about the target DNA's location in the original plate without disturbing or damaging the original plate. That's why the replica plating process is very, very important. Okay, so this is how the screening of genomic library is done. This is a very simple process of screening that is discussed, but in reality the screenings are more complicated. In fact, the construction of genomic DNA library is very easy, but the maintenance and screening is the difficult part of a genomic DNA library. It's a so difficult part. That's why people don't focus on this process. So screening takes longer time than construction. So that's all about genomic DNA library construction. I believe you have a clear idea about the genomic DNA library, how it's done, what is the importance of genomic DNA library. If you like this video, please hit the like button, share this video with your friends, and subscribe to our channel to get more videos like that in future. Thank you. Bye.